All right, welcome back, everyone. So I'm going to introduce the speakers for our next series of presentation. It's going to be a series of case studies to showcase you the efforts that C-Path rare disease activities, whether through RDCA-DAP or through public-private partnership, are doing to help accelerate drug development in their respective space. So we're going to have a series of presentations with a focus on endpoints and disease modeling development and how it can be used to inform drug development. And our RDCA-DAP works as a facilitator to help this effort. So for our first speaker, I'm going maybe to welcome to the stage my colleague Amanda Klein. There she is. Amanda is our executive director for the Therapeutic Transplant Consortium. She leads a multidisciplinary team to help advance product development for transplant with a focus on kidney transplant and long-term graft survival. Amanda is a fellow PharmD with direct experience in inpatient settings. So Amanda.
Thank you. All right, can you hear me okay? Great. All right. Well, I am grateful to be here and lead the case studies. So let's get to it. I'm going to be talking about the iBox scoring system, which is a composite biomarker that we are seeking regulatory endorsement. Yes, ma'am. That way? There we go. I just got to push really hard. No disclosures. Okay. The Transplant Therapeutics Consortium was formed in 2017 to accelerate the medical product development process in transplantation. This is accomplished by convening stakeholders from diverse settings, including industry, academia, and regulatory agencies in a pre-competitive collaboration. The current unmet need is developing novel immunosuppressive therapies targeting long-term graft survival. And our current efforts are geared towards long-term graft survival, specifically in kidney transplantation. Okay. Graft failure has significant negative outcomes, including return to dialysis, decreased quality of life, retransplantation, and then subsequent decreased donor pool. Graft survival is the most important outcome for people living with a transplant. Currently, therapies have drastically improved short-term outcomes with the one-year graft survival rate hovering around 91%. But the challenge for sponsors is executing registration trials of a reasonable size to support superiority claims for 1 to 2 year studies. And then when we look at long-term graft survival, this remains suboptimal. The 5-year survival rate in primary kidney transplants in the United States hovers around 79%. And the challenge here for sponsors is designing practical trials with a 5-year primary outcome. The currently accepted endpoints are not sufficient to assess novel immunosuppressive therapies aimed at improving long-term graft survival. Moreover, given that the causes of late graft failure are multifactorial, predicting such failure accurately with a single marker may not be optimal. A critical path innovation meeting, a CPIM, was held in which FDA expressed agreement on the unmet need in transplantation and supported the qualification of a composite score as a reasonably likely surrogate endpoint for use in support of drug approvals through the accelerated approval pathway. In 2019, the Paris Transplant Group, together with members of the transplant community, published a seminal paper on the iBox scoring system. The iBox is a risk prediction tool that utilizes multiple clinically relevant features that have demonstrated to be mechanistically associated with increased risk of late graft functional decline and failure. These include estimated glomerular filtration rate, eGFR, proteinuria, donor-specific antibody, and biopsy. These measures are taken cross-sectionally at any time point post-transplantation, and then these measures are integrated to generate individualized predictions of outcomes at 3, 5, and 7 years post-transplant. The iBox scoring system, when it was developed, was meant to be used at the patient level to inform clinical care and management of transplant patients. Data was prospectively collected from 4,000 subjects across 4 health centers in France, and this was used to develop the iBox scoring system, and then it was externally validated in cohorts from the US, EU, and 3 independent phase 2 and phase 3 clinical trials. The iBox scoring system outperforms any of the single components of the iBox scoring system, and each component of the iBox scoring system has established clinical and analytical validity. The iBox scoring system in the Lupi et al. British Medical Journal 2019 publication was designed, like I said before, to be used at the patient level to inform clinical care and management of transplant patients. We have adapted the iBox scoring system to be used as a clinical trial endpoint, incorporating feedback from both FDA and EMA. These optimizations include risk evaluation is fixed at 1 year post-transplant. This adaptation is based on feedback from EMA, noting that time from transplant is an important prognostic marker, but is never affected by therapy. Therefore, having the risk evaluation fixed at 1 year post-transplant does not impact the output of the model. Follow-up time was set at a maximum of 5 years post-transplant. Subjects who die, lose their graft, or withdraw before the first year of transplantation have missing iBox scores because it's before their 12-month evaluation. Therefore, analyses were performed imputing a 1 year iBox score for these subjects to allow the iBox scoring system to be used in de novo clinical trials as a primary endpoint. The imputed iBox score corresponded to a worst case scenario. In other words, the worst iBox score, and this was to prevent survivor bias. And then lastly, in the qualification submissions, we included 2 models, one model with biopsy input called the full iBox scoring system, and the other model without biopsy input called the abbreviated iBox scoring system. The iBox scoring system utilizes 4 established clinically relevant patient features that are routinely collected in transplant centers and clinical trials to compute an iBox score at 1 year post-transplant for the 5-year risk of death-censored graft survival. These features include eGFR and proteinuria that are measures of kidney function, presence of DSA using a qualitative binary mean fluorescence intensity cutoff, and then with or without kidney biopsy data using Banff lesion scores. Shown on this slide is the abbreviated iBox scoring system, so without biopsy input. The iBox scoring system is a composite score with a linear combination of the parameters from the multivariable cost proportional hazard model. The component measures were assessed at 12 months post-transplant and used to compute the iBox score. Because this is a composite biomarker, obtaining the data sets with the necessary variables to calculate an iBox score was challenging, as expected. For example, urine protein to creatinine ratio was not the proteinuria measurement used consistently across data sets with all the necessary variables. And so analyses were conducted supporting the conversion from various proteinuria measurements to UPCR for use in the iBox scoring system models. Another example is DSA MFI. The assay was granted 510(k) clearance by FDA as a qualitative test. The assay, so each tissue typing in user laboratory determines their own laboratory specific cutoff for presence of antibody, and then validates this cutoff according to the manufacturer's package insert for that assay. The optimal cutoff was 1400 based on literature precedent. Thus, we were able to align the analytical intended use of the assay with the clinical utility. And then lastly, we collected the assay information and laboratory certification documentation from the data sets to support the composite biomarker panel. We reviewed the documentation and deemed that the analytical methods were robust, reliable, and fit for purpose. On this slide, you can see that each component of the iBox scoring system, this one's specific for the abbreviated iBox scoring system, measured at 12 months post-transplant. This table shows the relative contribution of each factor in the model for the abbreviated iBox scoring system. We are working to qualify the iBox scoring system with both FDA and EMA. We have an accepted letter of intent in FDA biomarker qualification program and submitted the qualification plan, which is step 2 of a 3-stage process, in April, which is under active review by the agency. We submitted the briefing package to EMA for a qualification opinion in February and anticipate the decision in October, so next month. This is the first tool of its kind to seek regulatory qualification for use in kidney transplant clinical trials. You can see the proposed context of use currently presented to FDA, and this supports a quantitative basis for the iBox scoring system at 1 year post-transplant is a reasonably likely surrogate endpoint for the 5-year risk of death-censored allograft loss. Accelerated approval and the reasonably likely surrogate endpoint are specific to FDA terminology. So there's 5 data sets that were included in the qualification submission. The derivation data set represents the 4,000 subjects from the Luby et al. Publication. And the validation data set consists of 2 transplant centers, Mayo Clinic Rochester and Helsinki University Hospital, as well as 2 RCTs, the Benefit and Benefit EXT. With those 2 RCTs, these represent the largest population of patients receiving non-standard of care therapy. So it's calcineurin inhibitor is the standard of care, and these populations represent non-CNI based therapies. So the point of this is to show that the iBox scoring system was tested in different therapies to make sure that it worked across different populations on different regimens. The predictive performance of the full and abbreviated iBox scoring system models were tested in the qualification derivation data set. The C statistic gives the probability a randomly selected patient will experience an event. So in this case, graft loss. And that it's a higher iBox score at 1 year than those who do not experience the event, graft loss. Both models have high C statistic values of 0.85, 0.84 respectively, suggesting they can discriminate accurately within the construct of the proposed context of use. And likewise, we tested the performance of the iBox scoring system for both models in the external validation, and they have good discriminatory ability as well across the data sets. The next couple slides will focus solely on the abbreviated iBox scoring system, but it's worth noting that the predictive performance is similar for both models. The predictive versus observed graft losses were not significantly different for the abbreviated iBox scoring system, indicating good calibration in these data sets. These models, the iBox models, have reasonable predictive performance across the qualification data sets to support the proposed context of use. The treatment effect was performed to demonstrate the iBox scoring system can provide the evidence from clinical trials that a treatment effect on the surrogate corresponds to an effect on the clinical outcome. This was performed on 3 data sets, the 2 RCTs, BENEFIT and BENEFIT-EXT, and a subset of subjects on a specific therapeutic regimen, which is mTOR based. The operating predictive and prognostic characteristics of the iBox score show that the lower the iBox score, the better, and is associated with a higher expected death-censored graft survival. The treatment effect on the iBox score, which is negative and statistically significant in all 3 trials, corresponds to effect on death-censored endpoint, which is also negative in all 3 trials and statistically significant in the BENEFIT RCT. So in a prospective clinical trial using the iBox scoring system, we would need to power to help ensure the 5-year survival is significant, but these studies were not powered for that. The iBox scoring system as a composite score is not intuitive. However, the individual components are understood clinical parameters in kidney transplantation and can be used to model a minimal clinically important difference for an iBox score. So for example, an eGFR difference of 5 to 10 mils per minute is considered a minimal clinically important difference for renal function. And this is a slide to illustrate this example that how a iBox score, iBox changes for difference in iBox components. And you see in this example, eGFR. So investigated the expected iBox score difference if eGFR values were changed and all other parameters remain constant. eGFR is a well-known marker of kidney function. With a difference of 5 to 10 mils per minute is a clinically meaningful difference that has been defined and published. So a change of 1 mil per minute in eGFR resulted in a change of 0.046 in the iBox score, assuming all other parameters remain constant. So a difference of 10 mils per minute in eGFR resulted in an iBox difference of 0.46. And again, a lower iBox score is better and is associated with graft survival. So really in conclusion and next steps, the proposed context of use provides a quantitative basis to support the use of the iBox scoring system for use as a clinical trial endpoint at a fixed landmark. Qualifying 2 iBox models with and without biopsy will provide sponsors and investigators flexibility in their clinical trial design, whether they would like to include that surveillance biopsy at 1 year post-transplant. And that we have a qualification plan that we submitted in April that is currently under review by the agency. And lastly, we submitted the briefing package to EMA for a qualification opinion in February, in which we expect to hear the final in October. So this is future impact, what the qualification opinion from EMA that we anticipate receiving next month. Currently, there's only 4 endpoints qualified with EMA. This will be the first endpoint qualified in transplant. Once the iBox scoring system is qualified, it will allow sponsors to demonstrate superiority of the novel immunosuppressive therapy compared to the standard of care at various time points post-transplant in both pivotal or exploratory studies as well as in phase 3 studies. And then in Europe, this will allow label claims and promotion of superiority of the new therapy. Unlike in the United States, conditional marketing authorization is not linked to surrogacy. But with that said, one of the key criteria for conditional marketing authorization in Europe is addressing an unmet need. And so showing superiority to the current standard of care meets the criteria for sponsors if they would like to pursue conditional marketing authorization. And lastly, this is the first step to enhance development of innovative therapies in kidney transplant. And once again, this is a labor of love and could not be done without the international transplant community, the data contributors, and the TTC C-Path team. And one thing that we are working on currently is thinking about how can we leverage the current data, but also take the lessons learned to build a larger kidney transplant database based on variables of interest, not just to support a regulatory submission, but that it's a tailored data collection form to be both retrospective data and prospective for future drug development. And so this is an effort that we are actively working on now. So anyways, thank you all for your time.